He's here.
Okay. बाकी सबको कहना कैमरास ऑन रखें. Hi, Helge.
Hello, how are you?
I'm fine. How are you?
Good.
So nice to see you.
Good to see you, yes. If you don't mind, I'll actually join with a second computer as well, because that allows me, in case there's any trouble, to troubleshoot this and see this the way you...
No problem at all.
Presentation. Okay. So that should be doing it.
So you're doing it from home, I think?
I'm sorry?
You're at home or in office?
I'm at home, yes. We're mostly in home office these days, thanks to Corona.
How is it going? Again, lockdown or?
Not complete lockdown, but sort of soft lockdown. So we're strongly encouraged to work from home, stay at home, and only go outside if really needed. And there's some limited opening hours for stores and so on. But so far, schools are open.
Mm-hmm. Good.
And mostly do their lab work a little less than we used to, but it's feasible.
Good.
I guess things are a bit better for you these days, are they?
Yeah, it was much better 3-4 months ago, but recently we had a spike where things started getting worse. So, but luckily the mortality in Pakistan is quite less. So people do get infections, but mortality is low. We don't know the reason. Most likely some kind of genetic variation or, excessive.
Yeah, and probably age also, right? I mean, obviously,
yeah, maybe.
Switzerland is a lot older on average, I would say.
Yeah, then that's true as well. It could be a more younger population. But schools are off. Just today, they started the online classes.
How long have they been off?
They were off. They did it in November. In November, they said physically you can't come to school, but online classes went okay. So they had then this winter vacations starting from 24th of December, and just today, all the kids were in front of computers for online classes.
How's that working?
I feel, looking at my kids, I feel they got more quickly used to this as compared to us, the older generation.
Yes.
So my youngest daughter, who is in nursery, she's more handy. She's feeling better online than if she had to wake up in the morning and then go to the school. Then another one is Hajra. She's 9 years old, and she's a surprise to me. For a whole day, she can sit in front of computer, finish her work, and which I feel bad because, kids need to move around.
Yeah, they do. And they need the social interaction and so on.
Yeah.
I mean, I saw it. We had a school closure between April and May. Both kids afterwards were like, oh yeah, let's go back to school. It's going to be so great. They really didn't like it. I mean, they did okay at home, but you could see the difference. It's really, things are missing. So fortunately here right now, schools are still open, and it seems to be sort of, they try to postpone it as long as they can, another school closure.
Mm-hmm. Although I have seen kids are more immune. I have seen two of my nephews, younger ones, having the infection, but after 2 days they were okay.
Yeah, that's the idea of it, that they seem to get less sick and seem to also spread it less and don't seem to be as greatly affected if they get it. So that's why here they're saying, okay, let's try to keep the schools open as long as we can. But depends.
The worst thing with us was at university level. They kept our labs closed for 6 months, which was a disaster. Oh yeah. That was a disaster. We kept telling them, we tried to educate them because we are in a private university, and they really did not, they were scared that, if infection spreads on campus, but we were trying to tell them, look, grad students, the PhDs and, the thesis students, they are very few. So we can keep running the labs. So that was for us as researcher, that was a big loss. We had very hard time managing the flies to keep them alive.
So someone was still allowed to come in and
so Najma is there, right there. You can see she is a senior PhD. She'll be finishing this year. She, together with her husband, he finished PhD. He's moving for postdoc to US Northwestern. So both of them, I'm grateful to them. They were coming regularly during these lockdowns. Once rather they got challan as well from traffic police because I think there were more people sitting in car than allowed in the lockdown. but they managed to keep the flies alive. So that's good. How are things at FMI? So you have change of director. Dirk is there now.
We have a change of director, a year and a half ago or so. I mean, now for 6 months, he's sort of permanent director, or no, I guess almost a year now. He's been the permanent director, and great things ahead. Hopefully, I mean, it's still a great place. When did you leave?
I left in 2003. It is definitely the greatest place I have ever worked in for science. The environment there, the learning there is something I haven't experienced in ETH or even other places I worked at.
No, it's still a very fun place to be. I mean, right? It's sort of low in hierarchy, and there's lots of exchange among the groups, and there's the stellar facilities. So continues.
The biggest thing I always missed was these combined seminars. Like we used to have plant seminars, then there was a cell signaling group, there were seminars. So that was the whole learning culture. And then, as you said, the flat hierarchy. There was no hierarchy. You could talk to any senior group leader, professor, and so much to learn. In our times, Wittek was there, and George Thomas was there, Brian Hemmings. Is Brian still there?
No, he already retired. I guess 5 years ago or so. So quite a while already now. So I mean, we've really changed, right? So I guess when you were there, it was it growth control? Yeah, I think.
It was growth control, plants. Then there was neuroscience. There were 4 or 5 groups. It was
so plants are gone, as you know, and growth control is now quantitative biology. So things have changed quite a bit, of course, also with retirement of some group leaders and recruitments of new group leaders.
So you still have this culture of central facilities, like, there used to be primer synthesis, sequencing. So NGS is in-house, or?
NGS is in-house, yes. I mean, of course, it keeps changing, right? We no longer do primer synthesis, of course, because it's so much cheaper to get this from outside, and it doesn't really require any expert knowledge. And we no longer have an antibody facility, but we have NGS. That's a big one these days. We have computational biology as a core facility. So these are, I guess, the more recent new developments.
I was recently thinking about NGS for microRNA. So how do you go about? So you isolate total RNA and then do microRNA NGS, or you have?
Well, what you do is you isolate total RNA and then you size fractionate. Okay. And then you only take small RNAs and you directly ligate primers to them. So that's a bit different from the tagmentation that you do with the long RNAs. You immediately ligate primers to the small RNAs.
So you do size fractionation yourself, or you send it to facility and they do it?
Depends. I mean, some people only submit the RNA, and some that sort of need to do it more routinely. They do everything until the library preparation themselves, because it's quicker than if you need to go through the facility. Or sometimes we have some modification that we need specific classes of small RNAs. Then people also tend to do this themselves, and the facility only does the quality control and the pooling for the sequencing.
And what do you use for size fractionation? How do you?
There's kits. I mean, you can do it on a gel, or there's kits that use columns to do that. So there's different kinds of way. The columns are, of course, very convenient, very fast, very high throughput. The quality with the gels is a bit better. You get more reads of the small RNAs and less contaminants, but it depends on what your project is. Of course, it also depends on how much money you want to pay for the sequencing, right? If you sequence a lot of stuff that's not what you want, you're wasting money. But
yeah. So I was actually having a small project with diabetics here in Pakistan. And we were thinking of, because diabetes is something I would say more like a lifestyle. A lot has to do with the genetic makeup of the local population as well. So I was thinking of finding something there in diabetics which may work as a biomarker maybe. And then wanted to see if they are regulated by hyperglycemia, etc. Slightly away from my mainstream thing, which I focus pretty much on flies and polychondriathorics. But recently, collaboration with a hospital, I did this. And then I was looking at different protocols, and I found that, we have to go through plasma. And then I was wondering, one has to, because there was a few people were suggesting, you send the total RNA. And I was wondering how do I get then my microRNAs enriched? And you highlighted rightly.
Yeah, I mean, it's possible. It depends on what services you have access to, right? I mean, it's possible that they may be able to do the fractionation for you or the cutting. I think it really depends on the arrangement that you have with them. But certainly it's simplified a lot since the beginning. So the early protocols involved like 4 gel running and cutting steps. So you can imagine how long it took to just get that stuff right, and how non-high throughput it was. So now there's sort of one column and that does the trick. So that's obviously making things a lot faster and easier.
Helge, I would also like to know, we wanted to schedule another session, you remember, where we wanted you to talk to students. And I think 2 weeks from now, our students will be, classes will start, and I think it will be good. So we will bother you again with, or maybe you can tell us later on at your convenience whenever you have date, because I think this time is good enough for you. It's early morning. So I would like not to miss that session, because our students in Pakistan, they will greatly benefit from that session.
Yeah, I'm very happy to do it. So definitely, I'll try to send you an email. If I do forget, feel free to contact me. I mean, no hesitation, right? I'm committed
Don't
to do
worry.
this.
Yeah, I'll do that. I'll do that.
Yeah. So I heard on the radio yesterday that you had a big power outage.
Yeah, there was a power outage in south of the province where we live. There was a technical fault, and people who were supposed to fix that, or the ones who were responsible, I think they overlooked that, and that took everything. So the whole power transmission or power energy sector is divided into three big sectors, three big regions. And the one in south of Punjab, there was this glitch. And I think they have suspended those 7, 8 people for overlooking and not appropriately addressing that right away, because then that took everything down. But as we are, so I saw in this power outage, the Twitter and Facebook was full of, we Pakistanis are quite light-hearted, or, full of jokes. More like Italians have quite many common things with us. so Twitter and Facebook was full of jokes, that often we have blackouts when there is military coup. So historically, and people were throwing jokes that, once the light will be back, we'll see change and blah, blah, blah. So, but luckily it was fixed till morning.
Yeah, that's pretty impressive, actually. I mean, I remember when I was doing my postdoc in the States, we had this blackout, and I think it lasted like 3 days or so till they were finally able to get back electricity everywhere. It was a major disaster.
Oh God! So we had it, I think, in last 2 years. This is second one, and I saw yesterday a tweet in which the blackout you are talking about in US. So one of my colleague at LUMS, she was mentioning that if there are blackouts in US, people come and vandalize street crimes, blah, blah, blah. And luckily Pakistanis were just cracking jokes. So things
that's a pretty good way to go with it. I agree. Much like.
Yeah. No, they fixed it quickly. And I think it was embarrassing for the government as well, because we have a new government and it's a very different government led by a former cricketer who was a captain way back in 90s, who won the World Cup. And he had a long struggle for, winning the elections. And since he has no experience, or they were learning on flight, something happens, they are on the defensive. Yeah. So I think it's time. Let me introduce you, and then we move on. So it's a real pleasure to have Professor Helge Grosshans. He is a senior group leader at FMI in Basel, Switzerland. FMI is a non-profit research organization affiliated with Novartis Institute for Biomedical Research. So Helge studied biotechnology at Technical University Braunschweig in Germany. And then he moved for his PhD with Professor Ed Hurt in University of Heidelberg, where he worked on RNA biogenesis and nuclear export in budding yeast. As a HFSP postdoctoral fellow, so HFSP is Human Frontier Science Program. It's one of the most prestigious postdocs. So he joined Professor Frank Slack's lab at Yale University, and he studied the developmental and molecular function of microRNAs in C. elegans. And his work, that was groundbreaking. I remember reading that paper. That led to discovery of LED7 microRNA as a tumor suppressor gene in C. elegans, and that discovery was that it regulates RAS. His work, then in 2005, he started his own group at FMI. I vaguely remember, I think I was in his interview talk, if I remember. And He is supported by competitive research grants from ERC starting and ERC advanced grants. And his lab focuses on microRNA function and mechanisms of developmental planning. His group has contributed many breakthroughs among many others. So his group was the first to demonstrate that animal microRNAs are themselves regulated through RNase mediated degradation. And they also discovered that in a reversal of normal function, messenger RNAs, they can modulate the levels of microRNAs that target them. Grosshans now focuses on characterizing the underlying machinery for microRNAs by combining theoretical and quantitative experimental approaches. So today, Helge is going to talk about properties and functioning of a developmental clock. Helge, it's a real pleasure to have you. I'm really grateful to you for sparing time. Over to you.
Yeah, it's a great pleasure to be here and to be sharing our work with you, which actually has nothing to do with microRNAs in this case, but all with developmental timing. And we'll take quite a tour. It'll be quite a number of different approaches and ways to look at data and to interpret results. If anything is unclear, feel free to interrupt me, to stop me and ask questions at any time. I mean, obviously we're a small group, so it's perfectly fine to have this somewhat interactive. Also, if you have some ideas or questions, please do not hesitate to interrupt. Okay, so as I said, we're interested in the function of a developmental clock. And of course, you're all well aware that timing is quite important in our daily life. And usually you notice when you miss a bus or something like this. But timing is also important in development, even though that seems to be less clear on our minds typically. So if you think of you want to generate a pattern, you want to build a body, it's not only important where a certain cell is going to divide or differentiate or die, but also when it's going to do this. And how do you keep track of time? How do you keep time? Of course, typically you do this using a clock in your daily life, right? But what we see here as the clock actually is only the surface. We see the arms moving around. And I should also say a different way of viewing a clock is to look at it as an oscillator, an autonomous, a self-sustained oscillator. So machinery that internally generates a rhythm. You may have to supply it with energy, a battery, say, but actually the repetitive pattern is something that the clock, through its mechanism itself, generates. Now, as I was alluding to, what we're typically looking at is the output of the clock. We're seeing the arms of the clock, and that allows us to read time. But as a scientist, because what we find much more interesting is to understand the mechanism behind it. So the clock work, how does this work? How does this generate a repetitive signal? How does this allow us then ultimately to read time? And by this analogy of looking at oscillators, the clockwork then can also be referred to as the core oscillator, the core machinery. And of course, what it'll have and what as a molecular biologist we would be interested in are the the components of this core oscillator. So for instance, these cogwheels here. But if you look at these cogwheels now, they don't tell you how a clock functions, how a clock works. What you need to know on top is how do these cogwheels interact? Or in more genetic terms, what's the wiring? How do they talk to one another to then generate these oscillations? Now, if we're looking at biology, I think you're all familiar with certain types of clocks and oscillators. So for instance, you've heard of the circadian rhythms or circadian clocks, also sometimes referred to as body clocks, that generate the rhythmic behavior, day and nighttime behavior. So for instance, sleep and wake cycles, but also temperature cycles in your body or gene expression cycles, such as shown here for collagen expression in mouse tendon. And you may also have heard about somitogenesis clock. Somites are blocks of tissues that are repetitively formed in the embryo. And from these somites, your vertebrae and ribs and so on, and the muscles are formed. So these are clocks we know in biology. But of course, in these, as well as in other cases, again, what we're typically looking at is the output. So something that is rhythmically produced, or a gene that's being rhythmically expressed. But as I said, what we would like to understand is the core oscillator mechanism, the core clock. And in these core oscillators, we know that certain motifs of interaction can generate oscillations. For instance, a delayed negative feedback loop here, or they can also be more complex, such as a combination of a negative feedback and a positive feedback loop, but they may be even more complex. Finally, then, if we look at the output, if we look at what's rhythmic, we can often infer some biological function. Also, if we take into account features such as the period, so the duration for one cycle. In the circadian clock, as I said, it's about a day. So what it does, it essentially allows us to anticipate cycles of day and light, or food and no food. But the biological function also requires that the oscillator has certain properties, and these will be determined by its components and the way that these components interact. Conversely, then, if we have a given architecture of given components, that'll generate certain properties, and those properties will then enable certain biological function. Just to give you a flavor of what I mean by this abstract idea, if you look at the circadian clock, we know that it's temperature compensated, meaning it runs the same at different temperatures. And of course, that's very important if you want to use it to anticipate geophysical cycles, because a day is 24 hours, whether you're on the equator or on the North Pole. So you need temperature compensation. By contrast, if you look at a developmental clock, in many organisms, the duration of development will actually scale with temperature. So if you go to lower temperature, it'll take the organism longer to develop. So that's a very different property of such an oscillator. Now, finally, then, as a bit of additional introduction in these oscillations, there's different ways of how we can look at them. I've shown you, oops, the clock type of oscillation. So you can see this here as a circle where you go around, but you may remember from trigonometry 101 that you can also look at an oscillation as a sinusoidal wave. The features are actually the same, it's just different coordinate systems. This is an XY coordinate system, this is a polar coordinate system. And in particular, we can look at a defining feature, that's the amplitude. So in this, as a sinusoidal wave, that's half the displacement from the mean. That's the maximum displacement from the mean. So half the absolute fold change. If you look at the cycle here, it's going to be the radius of the cycle. We also know the period, of course, that's the time it takes to go through one cycle. So for instance, in the clock, that would be 12 hours. Or in a sinusoidal wave, you go from trough to trough or from peak to peak. By definition, of course, we know that the cycle is 360 degrees, or if you put it into radians, that's 2 pi. And I'm mentioning this because I'll be going back to using these different numbers as we go ahead, often interchanging between them just for mathematical convenience. Okay, is that clear so far?
Yeah.
Okay, again, interrupt if things are not clear. Okay, so we're studying oscillations in the worm, and what you're seeing here is one trace of one mRNA, how it changes over time. So we can synchronize worm populations at the time of hatching by hatching them in the absence of food. And then if we supply food, they'll start developing synchronously, and we can extract RNA at different times. So here we've done this hourly. and measure the levels of mRNA. And you can see that this particular mRNA has these 4 peaks of oscillations, these 4 rounds of oscillation. And that's true not only for one mRNA, but actually here we've done a time course going through the entirety of larval development and into adulthood. And in this heat map here now, every line is one gene. And red signifies high levels of expression, blue signifies low levels of expression. And you can see that all these genes here undergo 4 rounds of oscillations. And that's a large number. That's almost a quarter of the larval transcriptome. So 3,700 genes have this oscillatory pattern. That's at a cutoff of 2-fold change from trough to peak. And the period is around 7 hours. It varies somewhat with larval stage. Okay.
So this oscillation takes place during first 7 hours?
So 1 round takes roughly 7 hours. That's the period. And then it goes through the entirety of larval development. So from roughly 5 hours is when it starts after hatching to 36 hours after hatching, when the animals become adult.
Okay.
Okay, so before we go more into this, let me just briefly give you a little bit of background into C. elegans. Why 4 oscillations? Well, if you look at the development of the worm starting from the embryo, you'll notice that it goes through 4 larval stages that are designated L1 through L4. So we essentially have 1 oscillation, 1 peak per larval stage. These larval stages are separated by molds. So these worms, C. elegans, they have an exoskeleton made mostly of collagens. And at the end of each larval stage, they synthesize a new cuticle and they shed the old one. And that happens during a time that's called lethargis, because the animals don't move much at the time and they don't feed. And that's something we'll exploit in essays that I'll show you later on. So another way then to look at C. elegans development is sort of in this call type pattern, where we have cycles of growth followed by molds at each larval stage. Now, to give you a little bit of additional background on C. elegans, why it's actually a great model organism to use, is it's very simple. It has fewer than 1,000 somatic cells, and the adult worm is about 1 millimeter in size. So you actually need a microscope to study them. It still has several well-defined tissues, such as gut, muscle, skin, and it has what we call an invariant and fully known cell lineage. What that means is that if we start from a fertilized oocyte, this will develop in a very stereotypic manner, undergoing specific divisions and differentiation events at specific times during its development. So we essentially know where each cell in the adult worm comes from, what its progenitors were. It has a short generation time, so it takes about 2.5 days at 25 degrees to go from an embryo to the next generation. It's cheap in maintenance. We feed it bacteria on agar plates. It's transparent. You don't see this in this EM, of course, but we can look at it by microscopy, as you'll also see later on. We can perform genetic experiments with them, because typically they're hermaphrodites, but we also have males, which allows us to cross mutations together or transgenes. And RNAi and genome editing work quite efficiently in this animal. So all of this together makes it a very powerful in vivo system to really study physiological developmental events. And if we look at where we find C. elegans, is actually quite broadly distributed, because it's been sampled a lot in Europe and in the Western US, but you can see all these green dots in many places of the world where it's been found. Of course, there's some places where it hasn't been sampled a lot, but it may well exist there as well in the wild. And we do find it in places like this, which doesn't look very appealing. What this actually is a rotting apple. So they like to live in orchards, such as this one shown here, in the rotting fruit where they eat the bacteria that develop on this fruit. Okay, so let's now go back to how we use C. elegans in the lab, and specifically how we study oscillations in this animal. Now, I showed you this heat map to look at every gene individually. There's a different way of how we can look at these oscillations, and that is we can look at one time point and all the genes together, condensing them into one point. And it's sort of, so we're looking at the RNA profile at this one time point here, 1 hour after hatching. And we asked how similar is the profile to the next hour in development and the next hour. So we're calculating a correlation coefficient between two different time points, between the RNA samples from two different time points. And as you would expect, as you go along time in this X direction, similarity decreases because of course the animal develops. And so like your own 25-year-old is probably different from your 10-year-old, whereas your 11-year-old may be much less different from your 10-year-old. That's why as you go further in time, you diverge more. But you also notice that at some point you suddenly come back to this light gray. And that's because you're sort of becoming more similar again to this previous time point. And you can see that this has the 7-hour period that I previously referred to. So this is just a different way looking at these oscillations. In this case here, we're looking at every individual gene. In this case, we're looking at the whole transcriptome at a given time point. Okay, so why do we think that this oscillator functions as a developmental clock? As I told you in the introduction, and just by looking at what oscillates, we can often get an idea. And one way then to do this in our case is to look gene ontology term enrichment, although I'm typically very skeptical of this. It has lots of shortcomings. I think it's very obvious here if we look at what are the genes that are enriched among the genes that have oscillatory expression. I'll call them oscillating genes in the future. It's essentially protease activity and cuticle and molting. These are the really main terms that come up. And of course, protease activity is likely also related to molting because A, we know that collagens need proteolytic processing. B, we know that during the shedding of the old cuticle, we need to actually cleave proteins, and we need to cleave the cuticle itself by proteases. So that really seems to be an important function. And in fact, if we look at the patterns of collagen expression, and C. elegans has a lot of these, more than 100 cuticular collagens, we find that many of them have oscillatory expression. So if we have 25% of all genes, is actually 72% of the cuticular collagen genes. Many of these are just called col because by sequence, they look like a cuticular collagen. And we find that they typically accumulate, have their peak phase, so the time when the expression is the highest during the molt. But we have other ones that have specific names, such as dumpy here or squat roll, that seem to cluster at these specific time points. So the highest expression for these three genes is at this time point, and for these is at an earlier time point. The reason we think this is interesting is that these genes are named for the phenotype that they generate when they mutate these genes. So dumpy means short and fat, and the mutant worms indeed are short and fat compared to the wild type worms here. And what's been known from the literature actually is that these three genes here form a subcomplex on the cuticle, and these genes here form a different subcomplex on the cuticle. So that's in a way a means to organize functional subcomplexes. And similarly for these squat roll genes, what the phenotype that they cause is normally the worms will move in a sinusoidal fashion along the plate. These animals now twist around their axis in a helical manner because they have a twist in their cuticle. So what does this mean? Our conclusion from this is that oscillations are not just a means to express something transiently, like a signaling molecule, but they're also a means to allow just-in-time production of something to reduce complexity. So to give you an analogy, if you want to build a house, it's probably a pretty bad idea to deliver the windows at the time that you're digging out your foundation, because chances are the windows are going to be broken before you can actually place them in your house. And it's the same idea here. You have lots of different cuticular collagens, but they probably have slightly different functions, and you need to make sure that you incorporate them at precise times during development. And I'd like to point out that, I showed this slide at the beginning of my talk, that rhythmic expression of collagens, though in a circadian rhythm, is something that's also seen in animals. And it appears to be important actually for the regenerative and homeostatic activities of tendon. So if we want to look at these oscillations in the worm and really understand what they do in development, then probably sequencing is not the best approach, because there we only see RNA expression and we always have to look at a population. Ideally, we would want to look at the worms as they develop, and ideally not just at one, but at many worms at the same time. So Henrik Brinkmann in Göttingen here developed a system where he can culture animals in microchambers on a slide. And each of these chambers here is actually big enough for a single worm to roam around and move freely and feed and develop normally. But it's small enough that it fits in just one field of view of the microscope, so we can always find the worm back and we can track it over time. And what we can do now is we can keep these arrays of microchambers on a microscope for 2 days or more, and can repetitively observe the animal. So we image them every 10 minutes over these 2 days. And we can look at their development by doing bright field imaging or DIC. And we can look at gene expression with appropriate fluorescent reporters by using confocal imaging, fluorescent imaging. And then we can generate these traces based on the signal. What you see here is the gray traces are each individual animals, and then you see the blue, which is the mean of the animals, and the shaded area is the standard deviation. And I think the first thing you'll notice is how similar the development is across all of these different animals. So in this case, there's no synchronization involved, there's no scaling involved. All we do is we put an egg in one of these chambers, we let it hatch, and we set the time point 0 to the time of hatching. And you can see that you get this repetitive pattern. You can also see, or we can look now at when the molt happens based on these behavioral specificities of the worm and the cuticle shedding. And you can see that the molt seems to occur always at the same time for these different animals. So for this particular gene here, relative to the peak of the expression. So we can zoom in on this. And I told you that we call this here the peak phase of an expression, but in fact the phase refers to just being on any point of an oscillation. Okay, so we can have here the trough, and we can then define, for instance, the trough as minus pi, and the peak would then be 0, and the next trough would be plus pi. We could also call this 0, pi, 2 pi, but for convenience we call this now minus pi, 0, and plus pi, or minus 180 degrees, 0 degrees, plus 180 degrees.
Helge, I have a short question.
Yes.
Here. So these oscillations, the GFP is H2B tagged. So this is the...
Yes, so I'm sorry, I skipped over this. We can quickly go back. Oops. What we do is we take the promoter of a gene of interest that has oscillatory expression, and we use that to drive a coding sequence of GFP plus PEST. PEST is a sequence that destabilizes the GFP, that allows us to get better dynamics, because if the GFP is too stable, we wouldn't see this up and downs. And H2B is for nuclear localization, and we use that just so that it's easier to visualize in which cells we are looking.
Okay, thank you.
Yeah, sorry, I should have said this. Okay, so we can now quantify when mold entry and exit happens. And I told you we define the peak as 0. This is minus pi, this is plus pi. And then you can see that mold entry, the beginning of the red trace, happens slightly before the peak. Mold exit happens sometime after the peak. Final thing, you remember from your math, pi is 3.14, the number pi. So you can see here, it goes from 0 to 3.14, 3.14. And you can see that indeed, mold entry, the gray boxes here, happens very stereotypically slightly before the mold. And mold exit happens some way after the mold. So that seems to be that oscillations and molting are somehow synchronized. But are they really more synchronized than just randomly, or could there be two different timekeeping mechanisms? So could there be oscillations of gene expression, and could there be an additional clock that says the molting when to happen? Now to test this, we can go in a little bit of a mathematical modeling. We can ask, what variation would we expect to occur for a mold if it were uncoupled from the gene expression? Okay, so the cycle stands for our gene expression, and we ask, where does the molting occur? And we know how variable the molting is because we've looked at many different animals. And we can then ask if our observation corresponds to the same error here. And if we do this, and if we take the ratio, we would expect that if it's uncoupled, it should be 1. So we should observe the same error as we would predict in our model. The model here is based on error propagation. I won't bore you with the details, okay? The important part here is if it's 1, it's uncoupled. If it's below 1, it's coupled. And you can clearly see we've done this for 3 different reporters. We've done this at 2 different stages. Oops, sorry. And you can see that indeed the value is always below 1. So oscillations and molting are phase locked. They're coupled to one another, suggesting indeed that the oscillator can function as a molting clock. And we can use, conversely, now the molting as a readout for oscillator function, which is important if we try to think about how can we find factors that are involved in the oscillator, that are components of this oscillator. So how do we do this? And now it gets a bit more molecular biology, which is perhaps a bit easier to follow. Let me remind you that I told you that typically what we see is the output of an oscillator. But what we want to get at is the core oscillator. The problem is we have 3,700 genes with oscillatory expression. If we had to guess how many genes will be core oscillator, we would say, I don't know, maybe a dozen or a couple of dozen. So not a lot, probably less than 1%. So how do we find these genes? Now, first we can ask, do we understand how these oscillations are generated? And I showed you that mRNA levels oscillate, but we wondered whether these oscillations were generated at the transcriptional level. And the way to test this is by doing chromatin immunoprecipitation coupled to sequencing analysis, ChIP-seq analysis. So what we do is we immunoprecipitate RNA pol 2, which transcribes the mRNAs, and we do sequencing of the associated chromatin, and we ask whether we see rhythmic chromatin association of RNA polymerase 2. And these are the same genes here now for which we get a ChIP-seq signal and the mRNA sequencing signal. And you can see that indeed we see oscillations for both classes of genes. At both levels of analysis, suggesting indeed that oscillatory transcription is a key part of generating these oscillations. We can look at this also more carefully by going back to our transgenic animals that express this GFP past H2B transgene from the promoter of an oscillating gene. And what I haven't told you yet is that these transgenes are actually integrated in a targeted manner into the genome of C. elegans in single copy. So it should be quite physiological. And we can now do Rtq Pcr, so quantify the levels of this transgene over time and compare it to the changes of the endogenous genes, which the animals will express at the same time. And what you can see then, if you look here, the red is the trace for the endogenous gene. We have this oscillation over time. And the blue is the trace for the reporter. They actually overlap very, very nicely. In other words, saying that the promoter is actually good enough to recapitulate this rhythmic expression of this transgene. Again, suggesting that it's transcription, rhythmic transcription that drives these oscillations. So how can we then exploit this now to find components of the oscillator? Well, first I need to introduce you to an assay that we do to quickly screen through molting defects. I told you that the molting is associated with a period of quiescence where the animals don't feed, that's called lethargis. So what we do is we take animals that express a luciferase transgene and we feed them with luciferin. So this transgene is coming from fireflies. And if you provide luciferin, the animals will generate light that you can pick up with a plate reader. And we have here a multi-well plate, and in every well is one single animal. And so as we feed them luciferate, they'll continuously generate light, except during lethargis when they don't ingest the luciferin. And then we'll get a sudden drop in signal. And you can see this here. So red is embryo, the luciferin doesn't get through the eggshell. As the animal hatches, signal appears. And then as the animal goes into the mold, the signal suddenly drops. At the end of the mold, it suddenly goes up again. And the same again in the next mold, down, up again, down, up again, down, up again, 4 times. And we can do this in many animals, and we can again generate a heat map. Now every line is one animal. And again, this is sorted by the first entry into the first mold. You can see how stereotypic the development of these animals. Essentially, it always occurs at the same time for each of these animals. So what we can do now is we can manipulate transcription factors, in this case, deplete them by RNAi, and we can ask, does anything happen to this pattern of molting in the affected animals? And here's one example of transcription factors known as GRH1 or grainy head 1, which you may be familiar with from the Drosophila, of course, where it's a quite famous transcription factor. And you can see that this pattern here is severely disrupted. So this is mock, our control. You see these 4 moles. You don't see this when GRH1 is depleted. And here's a single trace. Again, 4 moles in the mock and only 2 in the RNAi condition. And then something bizarre happens. And what this bizarre is, you can see here in the time-lapse movie, I'm only showing you excerpt stills. You can see that actually this animal explodes through its head because it doesn't form a proper head cuticle. And that's actually already quite interesting because grainy head is termed grainy head because it causes this weird head cuticle in the developing fly. So there seems to be some similarity here as well. So this GRH1 itself actually have rhythmic activity. At least it does have not only rhythmic mRNA levels, but also rhythmic protein levels. As you can see here is a Western blot. So we extract protein from synchronized populations of animals hourly. And you can see the levels of the proteins are high at the beginning of the mold, and then they decrease, and then they go up again, peak slightly before the mold, and go down again. We can also endogenously tag GRH1 with GFP and then look at its level in the developing worm by imaging. And again, you can see there's a peak slightly before the mold in each of these 4 cycles. This is one animal, and this here is looking at the average of 3 animals. You can see that the pattern is quite reproducible. Finally, then, we can deplete GRH1 at different times and ask whether it's indeed repetitively required for each of these molds. And the way we do this is we fuse the endogenous GRH1 by genome editing to an inducible dagron, the so-called auxin-inducible dagron. So if we add now auxin to the animals, GRH1 will be depleted, and this occurs quite rapidly, within much less than an hour. So we can add auxin now at the beginning of development, and you can see these animals go through one mold here and then nothing. So it's required to execute the first mold properly. We can add auxin after the first mold, then they go up to the second mold. We can do it for the third and the fourth, and you can see every time we fail at the next mold. So indeed, GRH1 is required repetitively. And without much going into the detail of this experiment here, we used now an asynchronous population of worms. Again, every line is one animal. And we add auxin at one given time. And because the animals have hatched at different times, they'll all experience auxin at different times. And you can see up until this point here, if we add auxin early enough before the mold, they'll arrest at the next mold. If we add it later, they'll progress through this mold and into the next mold, apparently unperturbed for this round of larval development. In other words, there's evidence also for rhythmic activity. Okay, so this looks very promising. Does something now also happen not only to development, but also to oscillatory gene expression? And our experiment here is the following. We let the animals progress through the first larval stage and beyond the first mold. And then we add auxin to rapidly deplete GRH1 to levels that are actually, and we've tested this by Western blot, to levels that are below the trough during normal physiological expression. And we do hourly sampling and RNA sequencing until the time that these animals stop developing. And what we then see is genes like this, LIP6, just an example. In vehicle-treated animals, we have this nice oscillation. In auxin-treated animals, we still have an oscillation, but the amplitude is actually much reduced. We look at this systematically. We see that a large number of genes change in expression. And the important part, without going too much into these heat maps, is that a large majority of these, more than 80%, are actually oscillating genes. So it appears that indeed GRH1 greatly affects oscillatory gene expression. The most exciting genes of those that are affected actually is GRH1 itself. We noticed that already 1 hour after we've applied auxin, that's the first time point where we sample, the levels of GRH1 mRNA are increased as we deplete the endogenous protein. And that is true throughout this time course. And that is interesting because it suggests negative autoregulation, which I alluded to at the beginning, is sort of a typical motif in an oscillator. We can now take a reporter where we use the GRH1 promoter to drive again expression of GFP and do imaging. And indeed, we see that also in this experiment, we can validate the effect. So GFP levels go up as we deplete the endogenous protein. We then perform ChIP-seq analysis, immunoprecipitating the endogenous GRH1 and asking, what chromatin does it associate with? And the largest peak of all was in fact in the promoter of GRH1 itself. And data I'm not showing you now. We found that this element here was essential for developmental function of GRH1 when we did promoter bashing experiments that were actually not guided, but done in parallel to the ChIP-seq experiment. So we do think indeed that GRH1 has all that it takes to be a core oscillator component, the first one that we've identified thus far. Now for the remaining 5 minutes or so, let me shift gears again and talk about something different, and that is how we can infer oscillator architecture and learn something about the properties and what they mean for development. So what is unusual about the oscillator that we study here in C. elegans is that we can actually see the oscillations start and we can see them stop. So if you look here at the first few hours of development, you'll see that there's actually a constant level of expression, not much changes, right? If you're red, you stay red. If you're blue, you stay blue. And it's only then that the oscillation starts. So now you go higher, lower, higher, lower. And the same is again true at the transition from L4 to adult. You can see oscillations occur and then they stop and you remain at a certain level. That's very unusual. That's not something you can see in a somitogenesis clock easily. That's also not something you can easily see in a circadian clock. And the reason that this is beautiful is that there's a lot of theory on oscillators. And specifically, there's this branch of nonlinear dynamics theory that's called Bifurcation Theory that deals specifically with qualitative changes in systems. So how do you go from oscillating to non-oscillating, or from non-oscillating to oscillating? And there's different ways this can be happening. But two are particularly characteristic. The first one is called a supercritical Hop Bifurcation. So as you go from oscillating to non-oscillating, what happens is that the amplitude of your oscillation gets smaller and smaller. And that's shown here in the black trace. That's the envelope of the oscillation. By contrast, your period will stay the same. So the distance from one peak to the next is going to be the same. And then at this transition here, at this bifurcation, you stop. No more oscillation. A different model is that of the saddle node on invariant cycle, or SNCC bifurcation, in which case actually the period, the amplitude stays constant. Okay, you're not changing the peak, you're not changing the trough. Your envelope is going to stay the same, the black line. But what happens is that the distance between the peaks gets greater and greater. So your period increases until it reaches infinity. Another way to look at this is here the oscillation dies, and here it actually arrests. It slows down to infinity. And this is something we can experimentally test in our data. So we can ask, if we look at these different cycles here of oscillation, does the amplitude change? Does the period change? And as you can see, if we now quantify the amplitudes and we correlate them from one cycle to another, they actually always fall more or less on this line, meaning there's no change in amplitude. If we do what's called a Hilbert transform to get at the period over time, we can see that initially it's quite stable and then it increases. So this is precisely what this model here would predict, the SNCC bifurcation. Okay, now we've put a name on this. This is actually help us to understand the biology of what's going on. And we would argue yes, because each of these models has different requirements and predictions. So if we look at a Hop bifurcation, we know that it works with a simple negative feedback loop. So a gene may just negatively regulate itself. It requires some delay. Whereas for SNCC bifurcation, we need a more complex setup. So that's already interesting if we think of models of how this oscillator may work. The immediate more interesting prediction, however, is about how these oscillators behave if they are arrested. Now I remind you, we can look at these oscillators also as circles. We go around the circle. So this black circle means a stable oscillation. We also call this a limit cycle. And stable because if you perturb it, every trajectory will lead you back to this cycle. Now, if you go to the stationary state, what happens is that this limit cycle, this oscillation disappears and you spiral into a stable fixed point. And I remind you, the radius corresponds to the amplitude. So the spiraling in actually shows you that the amplitude is reduced. You also notice that this non-oscillating state, this fixed point, is actually distinct located in a different area from where the limit cycle is, the circle of oscillation. By contrast, if we go to the stationary state in a SNC bifurcation, the fixed point actually arises on the limit cycle. So we're kind of arresting on the cycle, and it's always the same spot. Okay, so this all sounds very abstract. I realize this, but why is this important? Now, if you think you want to arrest development, presumably if you want to restart it, you want to restart it from where you left off. And this is exactly what this setup allows you to do. Whereas this setup, if you restart it, you may end up at any place on the cycle. So you cannot do this. In other words, this setup here allows you to have a developmental checkpoint. Is this relevant? Yes, we think it is, because we know if we go through development and we starve the animals, they'll actually keep developing till a very specific point. So they're not just going to continue developing till they run out of food. They're going to develop till they've reached the next mold exit, and then they arrest. So there's a nutritional checkpoint. Is this linked to what we see here? We tested this by initiating starvation of the animals 4 hours before mold exit. And we would have 4 predictions if our model was right. Fed in the starved animal should finish the mold at the same time because they would continue oscillations till they've actually reached this point here. Oscillations themselves will continue for several hours upon starvation. So again, there's not going to be an immediate effect of the starvation. And the oscillation will arrest, as will development, exactly at this point here following mold exit. So we looked at these animals, and indeed, we can, I'm not sure how well you can see this, but after the same amount of time, 13 hours, we could see the cuticle in these starved animals and in the fat animals. So this first prediction bore out. If we now look at the heat maps, this is continuously fed. You've seen this many times, but this is a separate experiment now. You can see 1 cycle, 2 cycles, 3 cycles, 4 cycles, all as previously. If we starve and we initiate starvation here, you can see that the oscillation initially continues. So you're getting more red here, getting more red here, more red here. Oops, sorry. Or more blue here. And it's only at this point roughly where you stop the oscillation. And you can see that you stop the oscillation because you have the segment here where suddenly this red area expands. Now, this is why I initially in the introduction showed you this pairwise correlation plot, because here it's actually easier to see. Again, let me remind you, in normal development, after a few hours oscillations start, you get black, more dissimilar from what you were initially, go back to gray, more similar to what you had initially. If we do this on the starved animals, you immediately see the big difference. You see there's this large white blob here. But you also see that initially, again, you continue to oscillate. So here's where we start starvation, but you're getting more and more and more black, and you're in fact getting gray again, and then you stop in the gray. Okay. Finally, then, we took separate samples of synchronized animals that we specifically harvested at the time of mold exit or mold entry, but here only mold exit for us is interesting. And you can see that they have the highest correlation to the time of arrest here. So indeed, the oscillations arrests in a state that resembles mold exit on the limit cycle. Okay. I remember some of this, I understand that some of this has been quite abstract. So let me break this down into a few small take-home messages. The first one is we found that thousands of genes oscillate in developing C. elegans larvae with high amplitude. We showed that this is the manifestation of a developmental or a molting clock. We identified a transcription factor GH1 as a core clock gene. And we showed that the oscillator architecture enables a recurring developmental checkpoint. where do we go from here? We want to identify, of course, the complete set of core oscillator genes. We want to understand how they are wired. And we want to understand the spatial organization of the oscillator. So at this point, we're treating the whole worm as one oscillator. But we don't know whether oscillations differ across different tissues. And if they do, whether the tissues directly talk to one another, or whether there's a core oscillator that tells all the tissues on which phase they have to be. With that, I'm done. I'd like to acknowledge a number of people. The initial work came from a PhD student, Gert-Jan Hendricks, who in studying microRNAs discovered, together with our bioinformatician Dimos Gaidatsis, this massive oscillatory gene expression in the worm. And then two heroes, really, Yannick Hauser and Milou Meuse, followed up on this and established many of the techniques that I showed you about. And additional people, Chiara, Katrin, Lukas, Smita, and Anka helped along the project. And of course, we're greatly indebted to our collaborator, Karisios Chairis, another group leader at the FMI who studies somitogenesis and with whom we've had many fruitful discussions and ideas about how to analyze our data. And our facilities, C. elegans facility, Iskra, our microscopy facility, has of course been very crucial, and functional genomics for a lot of sequencing. And we have financial support from a number of sources. And I'm very happy to take any questions that you may have.
Thank you very much, Helge. Wonderful. A new perspective and a beautiful talk. So questions? Grad students?
Yeah, so I have a question.
Yes.
In the initial few slides, you mentioned about the strategy. So what I get from that was you first performed the RNA-seq, and then followed by RNA-seq, you identified certain genes. And those genes you cloned under, the promoter of those genes were cloned and followed by GFP and H2B, whatever you mentioned for nuclear localization and else. So that was your strategy to identify the expression localization of genes?
Yes, so I didn't really talk so much about the localization, but of course that's also something we've looked at. So what are the tissues where these oscillations happen? And indeed, that's something we can look at from the microscopy. It's also something we've started doing by doing single cell sequencing analysis. And what we see is that essentially we see these oscillations in epithelial tissues. Different epithelial tissues. The skin is of course one of them, but there are additional epithelial tissues where we see this. Now the imaging, of course, itself doesn't tell us much about the functioning of the oscillator, so the core oscillator, but it does tell us, and hopefully will tell us more, how it's organized. So what we can do in the future, for instance, is we can try to perturb the oscillator in one tissue and ask what happens in another tissue. And that will tell us how tissues cross-talk to one another.
Okay, so you performed RNA-seq, you must have found many genes, out of which you validated grainy head. So is the grainy head being expressed through all the oscillations?
Yes. So I should say, maybe to briefly recap, yes, there's thousands of genes that oscillate. So it was key for us to have a functional assay that allowed us to look at genes that really have a role in this. And the functional assay here was looking at molting defects, having a phenotype that we can perturb. And grainy head, yes, has itself oscillatory expression, has 4 cycles of expression. And we showed that we require it for every molting cycle, and we infer from that also for every round of oscillatory gene expression.
Yeah, one last question. So in wild type state, are these oscillations constant? I mean, if there is no change in amplitude or time of the oscillation?
Yes, so when we look at, if we compare what happens in different animals, it's indeed very stereotypical. It looks very similar from animal to animal. If you, of course, think about what happens in nature to these animals, things will presumably be somewhat different. So, for instance, if you have different food sources, the developmental rate will be different. So therefore, the period of development will also be different. The idea of having these developmental checkpoints is particularly important and interesting in C. elegans because we know it sort of lives like boom and bust. Okay, I mean, there's times where it has plenty of food because an apple just fell down and there's lots of bacteria developing, so it'll develop rapidly. But then at some point that food will be consumed and the animals will arrest because there's no food. And they'll have to wait till they get to disperse to the next source of food, for instance. So in the wild, of course, we expect there's going to be a lot of modulation on that. But left under sort of the luxury, ideal spa type conditions in the lab, it's very stereotypic and runs with a very constant amplitude and period.
Because that seems amazing that molting is, I believe, a state where the animals molt when they don't find enough food source available or the environment is not suitable for them to develop further. So that's the time that they molt.
No. That in the worm it's different. So in the mold, in the worm, the mold is really part of their normal developmental program, and they need to go through these 4 level stages. What is best studied in terms of no food, starvation, stress conditions is the so-called Dauer arrest. That's an alternative to the L3 stage of development. So then the animals will build a different type of cuticles that makes them more sturdy. So you can, for example, throw them into SDS and they're going to survive that. They're very resistant to desiccation. They can be maintained for many months, actually, without food. And then afterwards, if you put them back on food, if you put them on more friendly conditions, they'll resume normal development and mold into an L4 animal. So the mold actually is also very stereotyped in C. elegans, and it's not subject to environmental cues.
So what really is the time where you can freeze them or inhibit them from further growth, like stocks? How do you maintain the stocks when you don't need them for your experiments?
Yes, so we're very lucky, and in fact, so when I was looking for a postdoc, I was also interviewing with a fly lab, and when I learned that you have to keep fly flipping all the time, I was like, oh no, that's not for me, because the beauty in C. elegans is you can freeze them at -80.
Yeah.
If we don't need them, we just freeze them down, and we
At
can go back years later and. No, we freeze them as starved little ones. That seem to be the animals that are most resistant to freezing.
Thank you. That's all from my side.
And it's usually something that's very attractive to hear to fly researchers, but I know that many people have tried in the fly and failed. So it doesn't seem to be very straightforward to freeze fly, even fly larvae.
Yeah, we can't we can't freeze them.
Yes.
Other questions?
Yes.
So I have a question, Helge. So this core oscillator, if grainy head is a core oscillator and it's a transcription factor regulating itself as well. So after every oscillation, you think, did you try to develop a gene network or genetic network? You know, these are the same genes which are being regulated coordinately, or in different molting, let's say L2 or L3, there are different networks coming up.
Yeah, it's a great question. We've tried a little bit of this. It's not completely straightforward. The issue here is that many of the genes are going to be, again, output genes. And so these are the signatures that you get. For instance, that collagens will be up during the molt. But that's, of course, not what we're after, right? We're trying to get the core oscillator. So what are the few genes that talk to one another? This, of course, is not very easy to grasp with statistical methods, because there is only going to be a few genes around. What we've seen gratifyingly is, so out of our screen, we got 6 hits. The molting defect screen, we got 6 hits. And we saw, looking at ChIP-seq data that we had, and they're published outside, that actually these 6 genes seem to sit in a network where they sit at each other's promoters. And we have some evidence also for genetic interaction among them. So we do think that they make a network. Unfortunately, yes, through sort of genomics types of approaches, it's relatively hard or computational approaches is relatively hard to disassemble this network and understand what's going on. So we need to have a bit more guidance, have leads from functional assays to understand this.
So what I was actually trying to think about, whether genes which will be oscillating as a function of grainy head, they will be same in, let's say, molting number 3 as compared to molting 1.
Yes,
it's same clock.
Yes, I mean that's what we expect. We haven't validated this in great detail, but what we've done is our single worm imaging analysis actually looked at a different stage from what we did in the RNA-seq analysis. And we know that the genes that we tried to validate all validated. And indeed, that's the feature of an oscillator that we would predict, right? That it recurringly does the same thing. Yeah. Now, I should clarify, when we speak of GRH1, we think it's one component of an oscillator, okay? It's not that GRH1 itself makes an oscillator. In theory, that would be possible by a delayed negative feedback loop. In reality, of course, it's usually several genes talking to one another that generate this oscillator architecture.
Another interesting thing which we also came across with one of our genes is, so when you interfere with grainy head, depletion of grainy head using this Dagron system, you see upregulation of grainy head at transcription level. Of course, you do have you showed there is a binding site of grainy head. The biggest peak is in its own promoter. I was thinking, could there be another system where you know the cell realizes that I need a core protein which is missing in the system, and I need to pump its expression. I mean, it may not be negative feedback loop. I'm just trying to think in a different direction. Yep. It could not be a transcription level negative feedback, but some other systematic signaling going on which says, okay, this critical component is missing and now you need to push up. Because otherwise in wild type, it is present in the wild type as well. This peak is present in the wild type as well.
Yeah. Yes, I think you're right. Perhaps it sounds a bit too teleologic to me, in sort of the cell wants to do something, but I do think that, of course, built into the architecture could be something that actually results more indirectly in an increase in GRH1 level. And at this point, we can't say to which extent the increase is driven by autoregulation versus something that's more complex and involving additional components, perhaps even non-transcriptional mechanism. Mm-hmm. I should also say we have no evidence that it's a direct negative feedback. You could also come up with a different model. You could say maybe GRH1 actually positively boosts its own expression, but if it's gone, something else can bind in its place, and that something else just happens to be more potent. In that case, then it on the outside, it would look at it as if it would negatively regulate its expression, but in reality, it promotes its own expression, just not as much as something else that could bind in its own stead. But that's sort of a general challenge, I guess, in transcriptional networks, that often we don't really understand how activation versus repression really works. To ultimately figure this out, what we would need would be a very minimal synthetic system, right? Where we would go and essentially just take minimal GRH1 binding sites, put them into minimal promoters and sort of tweak those and ask what happens if we now modulate GRH1 levels or levels of the other factors that are supposed to interact with this. We're trying to chop down the promoter more to really get at these individual sites, but that'll take us time, obviously.
Abdullah, are there other questions from Facebook or anywhere, or I go ahead with another questions? Okay, so so another thing I was going to ask is, so between two oscillations or between two molting, when you have these gray boxes, you know, in the developmental phase, the beautiful box you were showing. So are these in different larval stages? I mean, my mind says they must be different, but are these same genes being expressed? Now I'm talking about not about oscillation, but the developmental toolkit. So you go through molting phase of development. Is it the same genes in cells, different cells? Because you have 1,000 cells. It's maybe easy to look at.
Yeah. So sorry, there's different classes of genes. So if we look at the oscillating genes, of course, it's the same genes that are going to be expressed from larval stage to larval stage. And I didn't really point this out, but many of these genes also do not peak during the molt, but peak outside the molt. So we don't think that oscillations are important only for molting. They're also important for other parts of the repetitive development, growth, probably, for instance. If we now look at other genes, we can see that there are genes that don't change much at all. They're just constantly present. And we have a third class of genes that we call the rising genes. Those are genes that keep up, monotonously increase over time. These are in particular germline genes, which makes sense because the contribution of the germline to the whole animal grows as the animal grows. But there are also some other genes there. So yes, there are different trends in the data. It's just that our focus has been almost exclusively on the oscillating genes.
So it means, to sum it up, so molting is function of oscillation, but not vice versa. So oscillation is normal developmental tool.
We think it may also be to some extent vice versa. So of course it's easy to imagine that sort of the rhythmicity of molting itself will have some knock-on effect on the expression of certain genes. So we think as we start looking into this, we will see all of this, right? We will see molting as an output of this gene expression oscillator. We will also see that the molting feeds back onto the oscillator. And one of the questions, for instance, is there sort of a reset button? So by analogy to the circadian clock, we have this endogenous clock that roughly keeps a 24-hour cycle, but only roughly. And the way that it's aligned to the actual day is through daylight. And what daylight does is so-called phase resetting. So essentially, every morning, your body checks, okay, what's the light level? Does it agree with the light level that I would have predicted? And if not, it shifts it slightly, it aligns it slightly. And the same may be true with the molting, right? I mean, clearly, this is a major developmental event in the life of the worm. And you could imagine, for instance, as the worm is in the mold, it doesn't feed. Now it sheds its cuticle, it starts feeding again. So immediately you have some sort of nutritional cue. And that may help you to realign again your whole oscillatory system. I mean, that's just a speculation at this point, but we think it would make sense. I would be surprised if the system didn't exploit it, because obviously it's important for the worm also to be very synchronous then across its tissues, right? Yeah. You can't have a partial mold. You can't decide that on your head you're going to mold and on your tail you're not going to mold.
Okay, Abdullah, are there any questions? Let me thank Helge. Helge, I'm really grateful to you for being so generous with your time. It was a wonderful talk.
Pleasure.
We all had fun, and we look forward to the second session sometime soon. I'll bug you.
All right.
Have a good day there.
Thank you very much. Have a good day. Bye-bye.
Bye-bye.